This is mitosis. This is the start of mitosis, which is prophase. In prophase, in mitosis, the chromatin condenses into chromosomes that remain randomly arranged in the nucleus. Additionally, in prophase of mitosis, the mitotic spindle begins to form. I know.
In metaphase, the mitotic spindle attaches to the sister chromatids and aligns them along the equator of the cell. The alignment of sister chromatids along the equator of the cell ensures that each daughter cell will contain one sister chromatid from each set of chromatids, and create two cells with identical DNA.
In anaphase, the centromeres split and the spindle pulls the sister chromatids apart towards opposite poles.
And finally, in telophase and cytokinesis, the nuclear envelope forms, the mitotic spindle disassembles, the DNA decondenses from chromosomes to chromatin, and the cell splits into two. The two cells are diploid cells with identical DNA. The process of mitosis is used within the body to replace dying cells and to allow for body growth.
Now to compare with meiosis. In the beginning of meiosis, prophase 1 happens. In prophase 1, the chromatin condenses into chromosomes, which then connect into sister chromatids, which then connect into tetrads with homologous chromosome pair. At this time, they are crossing over genes between homologous chromosomes because they are in tetrad form. If they were to switch with their sister chromatid, it would not create any change in the gene sequence. The crossing over allows for variation between each gamete cell. This is mostly random and happens on average once per each tetrad.
In metaphase 1, the tetrads of the sister chromatids align at the equator of the cell. This allows for independent assortment because each set of sister chromatids can go to either side. This process is completely random.
In anaphase 1, the maternal and paternal homologous chromosomes separate and move to the opposite poles of the cell. These sets of sister chromatids are independently assorted, meaning that the combination of chromatids set could be either maternal, maternal, paternal, paternal, maternal, paternal, or paternal, maternal. In this case, we have a paternal and maternal combination.
Through the process of telophase 1 and cytokinesis, the cell divides into two separate cells. In this process, the nuclear envelope forms around the chromosomes and the spindle disappears.
Prophase 2. In prophase 2, the spindle formed and the nuclear envelope breaks up.
In metaphase 2, the two sets of sister chromatids align at the equator of the cell.
In anaphase 2, the spindle fibers pull both the sister chromatids apart at the centromere towards opposite sides of the cell. The DNA brought to the poles have the same chromosomes, but are slightly different because of some chromosomes having genes that have been crossed over from other chromosomes.
Knock on wood. Yeah. In telophase 2 and cytokinesis, the spindle disappears, the nuclear envelopes assemble around the DNA, and the cell splits into two again, creating 4 non-identical haploid gamete daughter cells.